Hello, everyone, and welcome to today's webinar titled Antibody Therapy Against SARS-CoV-2, a Global Collaboration. This webinar is part of the Coronavirus Virtual Event Series. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Erica Ollmann Saphire, Ph.D. Professor at La Jolla Institute for Immunology. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Ollmann Saphire, you may now begin your presentation.
Thank you. I'm pleased to join you today. So I'm joining you on behalf of this international consortium, COVIC, the Coronavirus Immunotherapeutics Consortium. It was launched in March from the COVID-19 Therapeutics Accelerator, supported by the Gates Foundation, MasterCard Wellcome Trust, and others. And studies were subsequently expanded this summer by NIH and the GHR Foundation. So all of us around the world are praying for immunity in the form of vaccines to get back to any sense of normalcy and get back to work, get back to school, and get back to seeing our relatives. And while we wait for those to come out, we are cognizant that there will be some complications. The vaccines will not be in all places at all times. There will be people who are not vaccinated yet. Right now, that's all of us. There are people who can't be vaccinated and in whom vaccines don't take or just didn't last. What if immunity only lasts a year and coronavirus comes back in another year? And so there's another way in which we can help rescue some of those people in whom vaccines didn't take or didn't last or who haven't yet been vaccinated, and that is antibody therapy. So a major goal of a vaccine is to produce antibody, ideally neutralizing antibody, in the recipients. And vaccines would also induce innate immune responses in T cells. I'm going to talk to you about the antibody response. The hope is that by presenting a viral protein, a viral antigen, the spike protein, usually in the coronavirus vaccines, your immune system would be primed and have an antibody response at the ready. And then after several weeks after exposure, you have this antibody response on deck so that you can attack and clear any entering virus and prevent disease. Now, the vaccines have a variable response. Some people will have a stronger antibody response. Other people will have a weaker antibody response. In initial data, often it takes a shot and a booster, and it might be 5 weeks for the antibody to arrive. And this is what we need in order to achieve population protection. But there's another way in which you can get antibody right away and deliver it as a drug. And this is important for people who have become infected and are not yet vaccinated, or in whom a vaccine didn't take, or in preventing infection in someone like a frontline medical worker. And that's delivering the right kind of antibody right away as a drug. We call that antibody therapy or immunotherapy. And we have a lot of history with delivering antibodies directly, usually by IV, for diseases. We have very potent antibodies against cancers that can seek out and help destroy individual cancer cells. We have antibodies against infectious diseases. We have antibodies that help dampen autoimmune diseases. And of course, we're all familiar with antivenom. Antivenom is polyclonal antibody against the snake toxin. And you know, with antivenom, as with antiviral antibody, earlier is better than later. For antivenom, you want that shot of that polyclonal antitoxin antibody right away to save your leg and save your life after a snake bite. Same thing with antiviral antibody. In the first early stage of infection, it's better when you can knock down the amount of circulating virus. So an antibody is a natural human molecule. It is what you would want and expect your own immune system to make had it been given the opportunity. But by finding them, making them in GMP facilities, and delivering them by IV, you can give somebody the antibody they wish they had right away. It is a transient form of protection. It might last 3 weeks. You can engineer them to last about 6 months. And so it won't last forever, but it will get you through a crisis. And it's important that for some people with certain underlying conditions, it doesn't induce any lasting change in the immune system. It doesn't require a fully functioning immune system. It just gives you the antibody you wish you had right away. And it can be used to prevent infection or to treat people who have already become infected. Now, the antibodies that we have to work with to discover the ones that might be used as therapies are those that have been made in people that have already survived infection. So usually the very first survivors early in the pandemic donated their blood, and in their blood cells, researchers searched for the antibody molecules that would be the most effective. Now, there's a lot of possibilities. There are a quintillion possible antibodies in human sera. So that's a quintillion. It's 1 with 18 zeros behind it. And that's a lot of possible antibodies from all the sequence arrays. And within any one survivor, there will be a range of antibodies present against perhaps whatever they have been infected with or vaccinated against. A percent or a fraction of percent would be against SARS-CoV-2, the infection we want to find an antibody against. And so within that giant haystack, you want to find the very best individual antibodies against SARS-CoV-2, the needles in the haystack. And how do you find them? Well, you have to look into the array of what's available. So an antibody is shaped like a Y. It's got 2 arms. It's got a base. At the end of the arms is a combinatorial binding site. There are hypervariable loops that make the structure of that end different from every antibody. So that's your antibody. These would be foreign invaders like the virus that fit with a very special shape and charge complementarity into that end of the antibody that we call the fragment antigen binding. And that end is so precise, it operates as sort of like fingerprint recognition. So a particular antibody would recognize a certain site on a certain pathogen, and only that. They're very precise. So they're laser targeted for a particular pathogen. So this is what they really look like. This is a structure from my lab of a surface spike protein of a virus on the left, and then the Y-shaped human antibody on the right. And you can get a sense of the scale and how the antibody fits a particular shape and a particular part of that pathogen. So there are millions of possible antibodies, and we want to find the very best 1 or 2 that we might deliver as a drug. So you probably heard about convalescent sera being delivered to survivors. That's just the whole array of different antibodies right from one survivor into the veins of another. And that convalescent sera is quite variable because it comes straight from a person. So it'll contain antibodies against whatever they've been infected with or vaccinated with before. And some have very potent responses against the virus of interest, and some have weak. The goal of a monoclonal antibody therapy is just to find the very best 1 or 2 antibodies among the millions that are made, scale those up, and just deliver those, just deliver the needles from the haystack. And so the question is, if you're searching among those millions of antibodies, how do you find the very best 1 or 2 that are the best? Another question is, how do you define best? What makes them best? How do you determine in the lab what's going to work best in a human? What experiment would you do, or what feature of the antibody would you look for that's going to forecast protective function? And the other thing is that often we don't just deliver one antibody called a monotherapy. We deliver a mixture, and it's called a cocktail. 2 seems to be the magic number that we often use, sometimes 3. If you're going to find 2 antibodies to deliver together, one advantage would be that they hit 2 different footprints. And so it's harder for the virus to mutate to escape 2 things at once than 1 thing at once. And if you're delivering 2 things at once, you might as well find 2 things that are complementary to each other. Sometimes you can find antibodies that synergize, where the whole is greater than the sum of the parts. Maybe binding of one opens up the binding side of the other. So the pair together is better than you might expect either one alone to be. And if you're going to make a cocktail of antibodies, you're going to find a pair. What's the best pair? How do you know? How do we search among all the different antibody therapies being mobilized forward and decide what's the best? Right now, there are at least 75 different efforts around the world trying to discover and mobilize these antibodies as pre- or post-exposure therapy. Which ones are the best, and how do we know? Well, another variable is that the assays or the experiments you might do to try to evaluate antibodies can give different results in different labs. Even if it looks like the same protocol, the viruses they use could be a different passage number. They could be using them in a different cell line. The cells could be a different passage number. The resulting cells could differ in receptor populations on the surface, or the viruses being produced could differ in glycosylation state. So, a certain value of the ability of the antibody to neutralize from one lab might be different from that evaluated in another lab. And so if you are choosing antibody therapies to go forward into clinical trials in humans, how do you look among the playing field and try to find the ones that have the best chances of success? How do you compare different groups' competing efforts? Well, first we might talk about how we might use these antibodies, because how you might use them determines how you might evaluate them. So you would use an antibody therapy in an attempt to save lives here from an infectious disease, and you can use them for treatment or you can use them for prevention. For treatment, the best use is going to be in finding people in early stage disease, people with mild to moderate COVID-19 who are at risk of developing severe disease, say elderly people, people with underlying immune complications. If you can treat them with antibody early in the stage of infection, you can knock down the quantity of virus. You can reduce their viral load, improve outcomes of disease. Lilly's first trial results came out this week, and it looks like treatment with monoclonal antibody reduced hospitalization rates of people that had received the treatment. So that's good. It can prevent someone from progressing to severe disease and save lives. Another use of the antibody is in prevention. So people on the front lines, healthcare workers, first responders, high-risk groups like pregnant women, or to mop up a disease outbreak. Let's say there was a number of cases in a high-risk facility like a nursing home. You could treat the other residents and the staff with antibody to prevent them from being infected, and that's important when vaccines haven't yet been mobilized or in people in whom vaccines are less likely to take, like the elderly and the immunocompromised. So we would use antibodies in two different situations. The goal of the consortium COVIC that I'm going to tell you about was to evaluate all of the different candidate antibody therapies being mobilized forward to figure out which among them are best, what makes them best, and what assays best get us there. And our funding comes from different sources. It gives us different mandates. So our primary goals of this consortium to figure out what the best antibody is and why is a translational research goal. We're evaluating all of the molecules going forward for therapeutic efforts side by side under standardized assays, comparing them to each other in an apples-to-apples way in the same experimental conditions to figure out which one of them are more potent and what kinds of features they have. How are they differ from each other? Do they hit different footprints? So if the virus goes and mutates, which ones are still applicable? Our other mandate, and this is especially from the Bill and Melinda Gates Foundation, is to find those antibodies that we can mobilize to low and middle income countries. And so we're looking for those that are very highly potent. If an antibody is very potent, you don't need as many milligrams to do the job. If you don't need as many milligrams to do the job, what you can manufacture can go to more people. We're also looking for those antibodies of great stability so that cold chain is less of an issue, and easy manufacturability. You might have a molecule that looks absolutely fantastic in the lab, but if you can't make it at a useful scale to treat people, it's no help to you. And so while we are comparing all of the therapeutic candidates rushing forward to clinic, we're looking for those we can use to save lives in low and middle income countries as well. So our primary goals were translational. But with my academic hat on, while we have compiled the study, put together the study, while we have over 75 different partners aligned to figure out what the right antibodies are, and all of their antibodies together in one place, we can have the opportunity to really learn something. We can look at the whole array of different candidates being discovered, evaluated, and chosen by different groups and figure out the landscape of different ways in which antibodies can protect against the virus. If we have this larger data set of everybody's antibodies from all the different discovery engines, we can make a broader, deeper data set to understand what features of an antibody make one more protective than another. Let's say we could have do we find that at a certain site they elicit natural killer cell functions, at another site you can achieve the most potent neutralization. And then the other goal, while we have all of the different antibodies of the consortium and all of the different experimental labs of the consortium and the simultaneous human clinical data coming back, we have a chance to figure out which in vitro experiments best forecast with in vivo success. And that's a really unique opportunity for coronavirus. So for most infectious diseases, you discover something in the lab, you evaluate it in your plastic tissue culture dishes, and the things that you think are best based on whatever you're doing in vitro, you might try in rodents. And if they work in rodents, you might try them in primates. And if they work in primates, then you proceed toward human testing. Well, we have an opportunity now where the human testing is in the same year as our in vitro and early in vivo experiments. It's not 10 years later. So there's an opportunity for immediate feedback and information on our discovery pipeline. Do our in vivo preclinical models really forecast in vivo success? How about the in vitro models? Which are the features that are actually correlating with what works in humans? So we have a chance here to not only evaluate the candidates, but evaluate our whole research pipeline. So the goal of the consortium is to put everyone's antibodies and everyone's expertise in the same place to move forward faster and more completely with a better informed study. Why would you go to this effort of a very large-scale international consortium instead of looking at antibodies within just one wheelhouse? And the answer is that we think we can get broader, deeper information. Each individual discovery engine has their own set of assays and their own criteria. We might get different kinds of information by comparing different efforts. So an example of what we might learn by doing a broader study than just analyzing the antibodies from one group alone comes from the work that we did for Ebola virus at the outset of the major West African outbreak in 2014. Right before that outbreak, in 2013 actually, the Ebola virus field had this set of results. We had an antibody, a neutralizing antibody. I'm showing that to you in orange. That's just the FAB fragment, one arm of the Y. That neutralized beautifully if cell cultured, and it could inactivate the virus. That's what we thought that an antibody should be able to do. But it couldn't protect non-human primate models. And at the same time, there was a cocktail of 3 antibodies, and I'm showing you their FAB fragments in purple and in blue. Some of them recognize a disordered, heavily glycosylated domain that we know is at the top, but we don't know exactly the structure. This cocktail of 3 didn't neutralize virus in cell culture, but it did protect the non-human primate models. And so we have one antibody that neutralizes and didn't protect, and a cocktail that protected in the absence of neutralization. Now, we had thought that whether or not that antibody could inactivate virus in cell culture was the primary thing that we would want an antibody to do. And then sorting antibodies by neutralization would help forecast what was going to give us in vivo success. But it wasn't. So there was something different going on. Is it that the blue-purple set was protective because it was a cocktail? Maybe we just need to hit multiple sites at once. Or maybe it had something to do with a domain we call the FC. That's the base of the Y. That's the part that recruits immune cell function. You can see under the details, under the MB003 cocktail, that if you produced it in one cell type, CHO, you didn't get as effective protection as if you produced it in a different cell type, in Nicotiana. And the difference is the glycan put on the FC and how well it binds to FC receptors to recruit immune cell help. So maybe there was something we needed to understand about the ability of these antibodies to recruit immune cell help. The other possibility was that neutralization just wasn't enough. Maybe there was something else that the orange guy was missing. Or maybe we weren't measuring neutralization properly. Maybe the way we in the field had been evaluating antibody neutralization wasn't taking into account the right metrics or the right features or the right values to forecast and success. The other possibility is that the Ebola virus field had just looked at too small a number of antibodies at the time, and we had found the exceptions rather than the rule. But we knew that we as a research field needed to figure this out quickly. And so we formed a consortium, the Viral Immunotherapeutic Consortium, the VIC, which gathered the molecules of 43 labs across 5 continents, whether they were academic labs or industry labs or government labs. And we started with 168 monoclonals, but we eventually went up to studying 230. And we measured 32 different features of the antibodies side by side to figure out what are the different things that an antibody could do and which of them were more strongly linked to in vivo success. What were the best antibodies and why, and what experiment would we do to find them? And so as part of this, we wondered if we were measuring neutralization the right way. There's lots of different ways in which you can measure whether an antibody would neutralize in cell culture. So we compared different ways of measuring neutralization of Ebola virus in culture. One was actual Ebola virus, one was a BSL-3 model, another one was a common pseudovirus system, and they differed in the presence and absence of a secreted dimeric version and how the readout was. And so we took our 230 antibodies and we separated them by their epitope, where on the viral surface protein they bound, in these different categories, cap, head, mucin base. And then we evaluated every single one of these antibodies and their ability to neutralize in 4 different ways. Those 3 different systems, and also the percentage of viruses that are left unneutralized, the bit the antibody leaves behind. And we plotted each one of these antibodies. So you see in the blue box, those are antibodies against the glycan cap, and there's antibody number 1 and antibody number 3 and antibody number 5, all the way up to antibody 156. We could plot their ability to neutralize in each one of these assays, whether it was strong in blue, moderate in orange, or weak or not at all in red. And if you zoom in, for example, into the cap domain, you find antibodies like these that are sort of what we expect. They don't neutralize in any assay. All 4 boxes are red. And they don't protect an animal model, or the protection is kind of weak. That's what we're expecting. They don't neutralize and they don't protect. We have found other antibodies, like antibody 5 and antibody 8, that did neutralize, or neutralized most of the time, but still didn't protect the animal model. And then there are other antibodies, like these, that only neutralized the authentic Ebola virus, EBOV, didn't neutralize in other systems, and some of them were highly protected. And so that's interesting. There's something that the measurement of the authentic virus neutralization picks up that the other methods did not. And if you look across the whole data set of different antibodies against different sites and whether they neutralize in culture versus protect in vivo, we get these results. There are those that do what we thought antibodies ought to do. They neutralize in cell culture, they protect in animal models, and they protect because they neutralize, and some of these robustly neutralize no matter how you measure it. There are other antibodies that do things we weren't expecting. Here are some that neutralize, and sometimes they neutralize really potently, but they don't protect in the animal model. You're missing something. And here are antibodies that protect in the absence of any measurable neutralization at all in any system. So whatever it is they're doing, we're not measuring it in that cell culture model. And here's a set of antibodies that are protective, and they mostly don't neutralize. They only neutralize authentic Ebola virus, and sometimes they're kind of crummy at that. So they're doing something else, and the authentic Ebola virus is picking up some of that model. So if we look at the outliers, we can see what we can learn. And this is why it was helpful to do that large 43-lab, 230 antibody study. That sometimes a lab would have one outlier, and they just have one antibody that's weird. You tend to ignore it. But when you find 15 that all have something in common, then you have a body to study. And so that's what we gained by doing this broad trans-lab study. If you look at the outliers, which is those that neutralize and fail to protect in animals, or those that protect but in the absence of measurable neutralization, you see what's different. The ones that protect without neutralizing, they protect by doing something that isn't mechanical blocking of viral entry. They tend to inspire those effective protection mechanisms by the base of the Fc. So they inspire phagocytosis or natural killer cell functions. They inspire the immune system to come and destroy infected cells. And by destroying infected cells, those infected cells are no longer factories to make more virus. So it's that immune-mediated protection that let these guys be protective. And the ones that neutralized, meaning they could block receptor binding or block rearrangements of the glycoprotein required to enter cells, they could mechanically inactivate the virus, but they still weren't protective. What are they missing? The answer is they're weaker at inspiring phagocytosis or natural killer cell activity. So that told us we needed two kinds of things. We needed neutralization functions, and we needed Fc effector functions. And in fact, we had so much data from 230 antibodies and measuring lots of different features about them that we could rank order those features, and you could make a logistic regression model of all the different things we did, or things you could measure high throughput in vitro, which of them correlate with protection in the in vivo model. So a blue bar is a good thing, that's a protective feature. A red bar is a bad thing, that's a feature that links to not protection. A big bar is important, a strong correlator. Tiny bar that doesn't get off zero origin isn't very important or isn't a strong effector. So what do we find? We find that neutralization is, as we had expected, an important predictor of protection, and that the most effective way of measuring neutralization is with authentic Ebola virus, with the natural amount of the secreted dimer present. The next most effective thing, which we measured in the VSV system at BSL-2, was the fraction left behind, the number of viruses that the antibody couldn't access. If that antibody left some of those viruses on the table and couldn't wipe them out, that was a bad thing. That tended to lead to death. So an antibody that tends to leave a few percent of viruses unneutralized, they tend to hit sites that are variable. And that site might vary by the presence or absence of a carbohydrate, that in some molecules it's there, in some molecules it's not, and so the antibody could bind sometimes. That means there's always some virus that escapes that antibody. Or the epitope might change in conformation or structure. So sometimes the antibody was bound and sometimes the antibody was unbound. So those variable epitopes did not lead to effective protection. So neutralization was good, but it depended on how you measured it. Where the antibody bound and how tightly turned out to be not as predictive as we had thought. But there are lots of features about the Fc that did correlate with protection, the kinds of cellular functions it inspired and the kind of glycans attached. And then these internal ones were usually early or late antibodies, first generation, second generation measured. And so what we learned is that neutralization is indeed an important predictor of protection, and we should also think about what the Fc is doing as a primary criteria, not just let the Fc ride along, whereas exactly where it bound wasn't as important as what it did when it got there. And so we learned that we really wanted two different kinds of protection brought about by antibodies. We wanted mechanical neutralization. We wanted the binding of that FAB fragment to inactivate the viral glycoprotein in some way. And the antibodies that neutralized tended to bind within that black outline, epitopes like the yellow stalk, the red base, the purple fusion loop, things that existed on the receptor binding core of the glycoprotein. We also wanted in our antibodies the ability to inspire Fc-mediated protection, and that tended to happen to epitopes on the upper and outer part of the glycoprotein. That might just be geometric axis in the immune cell, the binding of the antibody. And so we think that we want to make an antibody cocktail or find single antibodies that could bring about both of these functions. And in fact, the human clinical data bears this out. Of the different antibody monotherapies or cocktails mobilized and tested in humans in the Ebola outbreak, they frequently pair a neutralizing molecule with antibodies that can bring about Fc-mediated protection, or the one in the center is a monotherapy that hits a sweet spot where they both happen. So this is the study that we were running for Ebola virus when we were asked to set up a similar analysis for coronavirus this spring. It was launched by the COVID-19 Therapeutics Accelerator. The goal, again, to evaluate all the therapeutics heading to human clinical trials side by side, help distinguish among them, and to make sure that we can find molecules to save lives in low and middle income countries. Now, an important part of the COVIC consortium, as we set up for the Ebola consortium, is blinding all the samples. And that is to make the study very fair. So the molecules come in under their real name, and they're all given a code name, COVIC-1, COVIC-45, COVIC-88. And by giving them a code name, nobody knows whose molecules are whose. And all analysis and decisions are just purely data-driven. All the samples come in. They're all quality controlled and anonymized. And then identical boxes of the antibodies are all shipped out to partner labs. So whatever it is, so that different labs are experts in different things. Some sequence glycans, others measure neutralization, others measure those Fc functions. Every lab participating in this multidisciplinary study takes a piece of the analysis in which they are experts, and they get the box set of the identical antibodies, all numbered 1 to 200. So they don't know what they are; they're just getting the data, and we put the data back together. By blinding the samples, we can also protect the investment in the intellectual property of the companies. So some of the antibodies come from academics, others from nonprofits, some from small biotechs, some from multinational corporations. They have invested significantly in discovering and mobilizing and testing these molecules, and a lot of them have, valuable intellectual property investment. And they wouldn't be able to participate if it wasn't protected in some way. And so the blinding of the study protects that investment. All of the companies know which antibodies are theirs. I, the PI, I don't know. They're just samples 1 to 200 to me. But the companies get back all of that data, so there's a benefit to them. There may be different kinds of experiments they didn't have, or different arrays of information they weren't able to get on their own, or they just might be able to find out where their antibodies land in the pack. They get all that data back, and they can use that for their own purposes and their own investigational new drug filings. There's a benefit to different companies. So the samples come in, they're blinded, they're allocated and distributed to all the different experts in the field that are each analyzing a different component or feature of the antibody studies. All of that data then comes into a database called COVIC-DB, and the database is publicly accessible. Because all the samples are anonymized, we can build a database that will be available to all the researchers around the world to look at this landscape of 200 antibodies against SARS-CoV-2 and find out where do they bind, what do they do when they get there, do certain features link to certain sites, which things link to protection, how do we compare the predictive value of the different assays that are being done. So that database is going to be an important and lasting feature of this consortium. And from the therapeutic candidates we're analyzing, we're going to look and see which are some novel combinations that play well together, or molecules of exceptional potency, stability, and manufacturability to make sure we can save lives anywhere in the world, regardless of the ability of the patient to pay for them. So the different sample analysis that we're doing includes measuring of binding constants to different forms and domains and regions of the spike. We're doing high-resolution epitope binning to put the antibodies into different categories. High-resolution structural biology to measure footprints and contact sites. Measuring the activity of the Fc in different ways and whether there's enhancement or protection. The ability of the antibodies to neutralize in different viral systems and cell types. The frequency, location, and likelihood of the antibody-antibody cocktail be escaped by mutation, whether or not they confer protection in animal models, and what novel combinations we might be able to make or optimize that don't yet exist. So the samples come in, aliquoted, distributed, blinded, sent out to all these different partners to each perform a piece of the analysis. The data goes into the COVIC-DB. The antibody owners can view their own unblinded samples and then the blinded data from everybody else, and the data is available. So here are the assays that we're doing. So my lab is structural biology. We do cryo-electron microscopy and X-ray crystallography to map structures of the different antibody epitopes. Georgia Tamaros and Dan Bettinger at Cartera, and Georgia's at the Duke Human Vaccine Institute, are looking at binding studies, how well they bind, constants, doing epitope binning. Nexalis is performing pseudovirus neutralization, and Alex Vacrea's lab at UTMB is performing authentic virus neutralization, the BSL-3. Galit Alter's lab at the Ragon Institute is using high-throughput system serology to profile Fc-driven activities of all the different anonymized antibodies. Then Alex Vacrea's lab is also looking at Fc function and the resistance of these antibodies to any form of antibody-mediated enhancement. Texas Biomed will look at the ability of these antibodies to confer protection pre- or post-exposure in different animal models. And we're also going to evaluate a novel animal model that is a mouse not only transgenic for human ACE2, but also the neonatal Fc receptor to try to see if this model can correlate with the half-life of these antibodies in humans, how long they last. That's important for prophylactic utility. Yoshikawa Oka's lab will look at the location and propensity of these different antibodies to be escaped by viral mutation. And working with him also is Betty Korber's lab at Los Alamos National Labs, who's keeping an eye on mutations that arise in the coronavirus as it transmits in human-to-human propagation around the world to see if there are mutations that are rising so we can see if the candidate therapeutics will remain responsive. And that's going to be important as we go through years or seasons of coronavirus infection and reinfection. And then Bjorn Peters at La Jolla Institute is going to gather and analyze all of this data and is building the COVIC-DB database. So I and all of the reference labs doing these experiments are blinded to the real antibodies name and source. It's just sample 45 to us. Operation Warp Speed and the program officers and our program manager are unblinded. And so if the contributor has a chance to call and say that data is or isn't what we were expecting, or we'd like something rerun, or can you tell us more? And the program officers can use the quality control data to make their therapeutic choices. The owners of antibodies, of course, know which ones are theirs. They can see the data as it's collected, and they can have it for their own purposes, and they retain all of their intellectual property. They can keep publishing and developing their antibodies as much as they wish. We very much want to see these therapeutics made available, and we want to see them made available 3 months ago. So we are trying to accelerate their discovery and mobilization efforts. And so it started as an idea in theory. Could we gather all the different therapeutic candidates and evaluate them side by side and really try to learn some lasting information to make sure that we can find therapeutic combinations we can mobilize anywhere in the world? But would people contribute? And the answer is yes, they do. And this is a 96-well plate of antibodies being shipped off to one of our partners. And we want to thank them very much because it's a tremendous willingness on the part of these scientists to step up and help solve a global health crisis and to share their therapeutic candidates, in which they've made tremendous investment for other people to go and evaluate. And the contributors thus far include academics, nonprofits, small biotechs, large biotechs, and major multinational corporations with antibodies coming from 4 different continents around the world. So what's our data so far? Well, it's just starting to roll in this week. So one question you might ask is, we know that results vary by lab. What neutralizes in one site might not neutralize in another. So does the data of the consortium agree with that from the antibody contributors? And so this is some neutralization results from the COVIC consortium. We're looking at a pseudovirus assay in the VSV backbone at Nexalis compared to results of a different pseudovirus on an HIV backbone reported by the antibody owner and neutralization of authentic virus as reported by the antibody owner. And we get results that are fairly similar. So they're all single-digit nanogram per mil. Other times, some antibodies match the neutralization of the authentic virus, but not pseudovirus results published by the individual lab. And so I think that different pseudovirus backgrounds grow to different titers and have different glycans. And so there may be differences among assays, but what we are measuring seems to correlate with our authentic virus data. And for some other antibodies, particularly those that neutralize only moderately, we're getting ballpark agreement. So for example, we got an IC50 on our VSV background of 6 micrograms per mil. That's a little bit better than the antibody owner who reported 15 and reported that it did neutralize authentic virus, but we don't know the specific value. When we look at our binding studies, we see a range of binding either to Jason McClellan stabilized hexapro spike antigen with and without a D614G mutation that's become fixed in the world, and affinity to just the receptor binding domain and affinity to the N-terminal domain. So we do have antibodies against the N-terminal domain. The different molecules tend to bind better to the trimeric spike than individual receptor binding domain constructs, but we have a range of affinity. We have high affinity antibodies. We have lower affinity antibodies. And the affinity does tend to track a bit with the neutralization. So if we separate this pile of neutralizing antibodies by their potency and neutralization, so that's the IC50, so they're more potent at the left, less potent at the right. By their affinity, tight binders are at the bottom, and weaker binders are toward the top. And we can separate the antibodies by those against the receptor binding domain in the dot, and a couple against the N-terminal domain in the X. If you look, we don't have a large sample of N-terminal domain antibodies, but if you look at the receptor binding domain antibodies on the bottom left, those of greatest affinity tend to be of greatest potency, and that makes a lot of sense. So if they bind very tightly to the receptor binding domain, they're probably adequately blocking the ability of the virus to attach to receptor. That correlates with their potent neutralization. So we have 2 N-terminal domain binders there. These are the most potent against the receptor binding domain. When we line up the first 93 antibodies analyzed so far by their Fc effector functions, you can see there's quite a range. There are some that have been made to be or are naturally superfunctional, in phagocytosis in the top, or other functions in the bottom, and there are others that are made to be effectorless to make sure they couldn't possibly elicit antibody-dependent enhancement or other functions. And so they correlate in different ways. And this is a way of looking at a pie chart representation of the different Fc-mediated functions of each one of the antibodies. And there's some that have, with the big colorful pie pieces, that elicit an array of Fc-mediated functions and strongly, and there are others, the empty gray circles or the tiny pie pieces, that are made to be effectorless. So we'll see how those perform differently as different populations. We can cluster them based on their ability to bind to different Fc receptors and mediate functions there. So that's just the first bit of data that's come in this week, and all of our experiments are in progress. We're still contracting for more antibodies, and there's going to be more data to come. This is the setup and the launch. And I'd very much like to thank all of our experimental partners for doing all this heavy lifting and analyzing all of these antibodies in great detail. So Georgia Tamaris, Betty Korber, Sujan Shrestha, Bjorn Peters, Dan Bettinger, Yoshi Kawoka, Alex Bukreyev, Galit Alter, three at Texas Biomedical, Luis Martinez, Jordi Torres, and Joanne Turner. And most of all, I'd like to thank my program manager, Sharon Schindel at Jolla Institute of Immunology, for the incredible effort of all of the quality control, all of the concentrations, all of the shipping, all of the coordination, and the communication, because I'm blind in this study. We very much welcome feedback, participation. We're still enrolling new antibodies, and you can find me at the email address at the bottom. And I'm happy to answer any questions.
Wonderful. Thank you so much, Dr. Ollmann Saphire, for your thank you for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question that you would like to ask, please do so now. You're just going to click on the Ask a Question box located on the far left of your screen. So I have a few coming in. Let's get started. Our first question, what's your opinion on using, and excuse my pronunciation, intravenous immunoglobulin, intravenous, thank you, immunoglobulin in COVID patients, and in what stage should it be used?
So there are two kinds of intravenous immunoglobulin. Probably the question asker means convalescent sera, and that's the polyclonal whole range of antibodies from a survivor, some of which are against SARS-CoV-2 and others are against whatever else that their immune system has experienced. And the data are still out on exactly how effective it is, and I think that is because of the variability. Some people have a very strong antibody response, others have a very weak antibody response. So if you've gotten stronger antibodies, you could expect to get better protection than if you've gotten weaker antibodies. A more potent and maybe reliable mechanism of protection would be the monoclonal antibody treatments, like the ones that I've described here. And that's not just taking whole polyclonal, all the different antibodies straight from blood, but picking out the one or two that are very best and just delivering those. So it's like a laser-guided missile instead of a barrage of BBs. And based on the potency of what we are seeing go forward, we were expecting success, and the first results presented actually do look successful, that there's able to prevent hospitalizations that might have occurred. So with antiviral antibody, earlier is better. You want to use antibodies that will target and inactivate viruses early in infection before they grow into too many viruses and too many infected cells. You would use an antiviral antibody earlier. Later disease, the acute respiratory distress, much of that pathology is an overactive immune response. So at that point, there's not as much virus around. So an antivirus antibody isn't going to be as helpful as something else that would treat that overactive inflammatory response. And so a good example would be like antivenom. You want the antivenom right away. You don't want to wait a week or two and see what happens with the bite. So I would say as soon as you know that you have been infected, if you have this positive test, or if you are have a known exposure and you're a highly vulnerable person, that's when you want to get the antibody treatment. And there are lots of clinical trials running on lots of different sites now. You can go to the clinicaltrials.gov and look at the monoclonal antibody trials, and they look for ones and what they're looking for. Some of them want to treat inpatients. Some of them are specially designed to treat outpatients. And so if you are feeling just a little sick or not sick yet, but you know you've been infected, that's a really good time to get that antibody to make sure that you don't get worse. Get it earlier. Don't wait and see what happens.
Thank you. And then the next one I think correlates with that one from the same person. What is your opinion about using COVID-19 hyperimmune serum obtained from animals or humans, and which is better?
So hyperimmune serum is another kind of therapy where you could immunize an animal over and over again, and then they would develop very high affinity antibodies. And those can be more potent than something from a survivor who's been infected once. The original forms were animal antibodies and animal serum, and so you would want to use them with some caution so that you don't get serum sickness or an immune response against that animal serum. Now, there are new technologies, like there are cows that are transgenic for human antibody genes, and immunizing these cows gives you human antibody, and so you would not have an immune response against those, and some of those have been evaluated against the related MERS, for example. And so those are great therapies, therapeutic options as well, and you might get those discovered or available perhaps earlier than some of the monoclonals. I do like the precision of the monoclonals. It's a very well characterized response. There's often high-resolution structural biology and very thorough characterization in vitro and in vivo of how these different antibodies behave.
Great. And does in vitro enhancement function of antibodies correlate with in vitro disease enhancement?
Right. So antibodies can neutralize, meaning they block infection, or sometimes they can enhance. And if they enhance infection, that means they're making the infection worse. The antibody has somehow facilitated uptake of the virus. That is more rare. It is well characterized for different classes of viruses like dengue or Zika, less so for viruses shaped like SARS-CoV-2. Typically, any neutralizing antibody, if you dilute it enough in a test tube and take it below the concentration at which it could possibly neutralize, you can make it look like it enhances in a test tube or in a rodent model. That sometimes but rarely can correlate with any enhancement of disease in the animal model. Viruses like, and I think that enhancement of disease is something that everyone is quite worried about and looking out for, but it hasn't been observed yet in the clinical studies for this virus. Where it has been observed is something like dengue. So there's 4 different kinds of dengue virus, and if you get infected with type 1, you make antibodies against type 1, and then if next year you get infected with type 2, your antibodies against type 1 don't bind the different type well enough to neutralize it. Instead, they might enhance it. And so you get dengue hemorrhagic fever, a worse outcome of dengue on your second infection with those enhancing antibodies. That has been less observed for other diseases. Vaccine enhancement was noted for very early respiratory syncytial virus vaccines when inactivation of the vaccine particles sprung all the surface proteins into the wrong shape, so they made the wrong kind of antibody. We have sophisticated structural biology tools now that actually see what the shape is and can confirm that it's in the right position. So that kind of misstep isn't going to happen again. So typically, enhancement occurs at a subneutralizing concentration, and although it is something that everyone is very much watching for, it has not seemed to have happened in convalescent sera or monoclonal antibody administration for SARS-CoV-2.
Thank you so much. I have a question coming in about that question that you just answered. Are in vitro enhancing antibodies disqualified for COVID treatment?
I would think they would be. I think people look at if a discovery engine makes 1,000 antibodies, they pick the two most potent, and they're so potent that they just don't enhance. All they do is neutralize. And then some antibody manufacturers have taken the tactic that we want a functioning FC, that's the immune recruiting arm, to mediate that immune cell help. Others have taken the tactic that they want to eliminate any possibility of enhancement, and so they've made that FC effectorless. They've made that FC unable to bind to the any molecules that could facilitate that uptake. But the molecules that are being evaluated for human therapy are so potent that they are quite unlikely to cause enhancement if given at any good dose.
Got it. Thank you. Is there neutralizing activity outside of the RBD, and how does this correlate with the prophylaxis in hamster challenge?
Mm-hmm.
Okay.
There are neutralizing antibodies against the N-terminal domain, and I expect there are other neutralizing antibodies against other sites that will be discovered in the fullness of time. The quickest path to neutralization seems to be that direct block of receptor binding, the receptor binding domain.
Got it. And then the next one I have, do you have any ethical concerns for vaccines that are being developed because of how quickly they are being developed?
A lot of the speed in the vaccine development is eliminating things that are like administrative delays. So one example might be, let's just assume we're going to have some vaccines, and those vaccines are going to need syringes and refrigerated trucks and nurses to administer them. So let's assume they're going to exist, and let's start figuring out our logistics about glass vials and refrigerated trucks and nurses. Other things are just eliminating administrative delays. So typically you might submit your packet of data, the review group would be convened, they'd meet the following month, they'd review the data, it takes a week to write the report that goes back to you, you have to jump all the hurdles for clear to do the next step. Well, right now you're writing up your data, it goes to the committee right away, they review it, there might be meeting every single week or meeting more rapidly by Zoom, and they get you the information back that night. Or they might say, okay, look, we like data A, B, and C. We still need to see experiment D. So set up experiment D and go ahead and order what you need to do step E while we're waiting for the results of D to come back in so you don't have delays. And if we like it, you can go right into E and not experience another month or two delay. And if we don't like it, well, then we'll pull the plug on D. So a lot of the speed is just reducing the normal number of delays. Everyone's working 7 days a week and 12 hours a day to make this happen. And another way of reducing delays might be, let's say you were looking for infection events in your phase 3 trial. So you have to vaccinate a lot of people and then wait and see who happens to get infected. Well, if it's a rare event, you might wait a long time to see it on a small number of people, but if you can enroll more people, you might be more likely to see those infection events and determine if they're protected. So it's those kinds of things, administrative barriers and delays and being strategic, that's giving us a lot of the acceleration here. And I think it would actually be unethical to not try to bring protection about to the world. There's a lot of effort to make sure that everything has been thoroughly vetted by safety. No one's compromising on safety in these things. What they are compromising on is sleep and weekends.
Thank you. When we actually have a vaccine, how will it be assured that all individuals have access to it? For example, those who are uninsured, and how long will it take for it to be available?
I don't know the answers to those questions. My expertise is more the molecular workings of how do you get the right immune response. So I don't know. I know there's been an effort by different health departments to try to figure that out and get that organized ahead of time and figure out how do you organize a priority. Maybe the people that are most vulnerable are top priority. Maybe the people on the front lines, the medical workers, are top priority. And how do you go and build your distribution networks? I don't know the answers to all those.
Got it. And then we have time for one more question. When do you predict a reliable vaccine to be available for COVID-19?
Nobody, I don't know. And I think every immunologist is being asked this question, and most of us just aren't answering because it's not available until we see all the data, and the data comes when the data comes. And I don't know. It's a wild guess. Spring, probably. Early spring.
Got it. Thank you so much, Dr. Ollmann Saphire, for your time today and your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 6 months until the March of 2021. LabRoots will alert you via email when this is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you again, Dr. Ollmann Saphire. Until next time, goodbye.
Thank you.